Hello everyone, and welcome to the June 2016 Soils Network of Knowledge webinar. I will introduce Mick and the latest update on the GRDC co-funded project that's examining the impacts of herbicide on soil biology and function. Mick's got 10 years of research experience in agriculture and environmental chemistry. He's previously worked on constructed wetlands for improved water quality, plant growth promoting microorganisms, abiotic stress tolerance in plants, and organic amendments for soil health and plant productivity. He's one of only 2 speakers to make a second appearance at SNOC, so good on you, Mick, for doing that. And as I said, Mick will be talking about herbicide residues in cropping soils and whether they pose a problem to plant growth. It's a talk he gave at a recent Soils Masterclass convened by New South Wales DPI and funded by the Crawford Fund. Thank you, Mick.
All right, thanks very much, Abby, for the invitation to give another SNOC webinar. So I actually gave a talk on a similar topic probably a year and a half ago now at the start of our GRDC co-funded project, looking at the impact of increased herbicide use on soil biological processes. So hopefully I can answer or begin to answer that question there on the title slide. Herbicide residues in cropping soils, are they an issue? Oh, there we go.
Great.
All right, so what are the potential issues? Well, we know that herbicide use has increased quite a lot since the advent or the move towards conservation tillage. So a lot of farmers concerned about erosion in particular and soil health and the damage caused by tillage have moved to minimum tillage and no tillage, so minimising the disturbance to the soil. But along with that, because cultivation was a primary tool for controlling weeds, there has been an increased use in herbicides over the last probably 20 years, and this is continuing as well. So obviously there's the intended impact that we want herbicides to have, which is killing target weeds. However, when herbicides are applied, they are often exposed, or the crop plant is also directly exposed to herbicides, but also organisms, non-target organisms in the soil. Now these may be free living, for example nitrifying organisms that convert ammonium through to nitrate in the soil, but they may be associative, so they may be for example mycorrhizal fungi that actually act in concert with the plant to help the plant acquire nutrients. Then there's also fauna within the soil that help to move soil around and help with organic matter turnover. So really, I think farmers are beginning to ask what is the impact of this increased herbicide use on these non-target organisms and also crop productivity. I think a number of farmers believe that there's been a slow yield decline, and questions are arising. Is this in part caused by the increase in herbicide use, and what part of that yield decline can be explained by herbicides and what's due to other factors? Now, I don't want you all to go to sleep at this point. If I saw a talk where someone threw up this slide risk assessment framework, I know I'd be pulling out the pillow and preparing myself for a good nap. But in this case, it's quite important, because what I want you to realise here is that there's 2 components to trying to work out the damage or the risk posed by herbicides to crop production. The first is the toxicity. So what's the inherent toxicity of the different herbicides, and what is the level at which a plant or an organism in the soil is either killed or has a decrease in growth rate or loses some of its functioning. And particularly in terms of soil biological processes, what we're looking at here is what level of herbicide will begin to inhibit functions delivered by organisms in the soil that provide services to the plant. For example, nutrient cycling or pathogen suppression. So that's one side of the equation. The second side of the equation is actually the exposure. So although we might know the level at which a particular herbicide inhibits a function, what's the exposure in the field? And this part here really although it sounds simple, is quite difficult to define, in part because we need to know a number of things. First of all, we need to know how much of a particular herbicide is being applied, but each herbicide will break down and dissipate in the environment at a different rate. So working out how long those herbicides hang around is another aspect that we have to get a handle on. Thirdly, not all of the herbicide that is applied will be bioavailable. That is, not all of the herbicide that reaches the soil will actually be accessible to organisms in the soil, or the plant for that matter. So when herbicides hit the soil, different classes of herbicides will bind strongly or weakly to the soil particles, where they are no longer available to organisms. Once we have a handle on these two things, we can then start to look at the risk and what's the probability of a function being inhibited to the extent that it will cause issues to the crop. All right, so following on from that, what we don't know, or what we didn't know until recently, is from an industry-wide perspective, particularly in the grains industry, which herbicides are actually being applied. This data here comes from a project that has recently concluded over in Western Australia called the Focus Paddocks Project, where the Department of Agriculture and Food in Western Australia monitored 180 different paddocks throughout the cereal cropping region of southwest Western Australia. And they did this over 5 years and monitored all manner of inputs and measurements and yield, rainfall, weather, and including herbicide application data. So in total they had records from 556 crops. Obviously they didn't get every paddock every year, but a total of 556 crops over those 5 years. And what you can see is that, as you might already expect, the most commonly applied herbicide is glyphosate, and that's applied almost once on average to every crop. As you can see here, it's applied almost 500 times. So on average, once to every crop. However, some crops won't receive any glyphosate at all. Others might receive 2 or 3 sprays. You can also see a range of different chemicals here, and in fact the total number of chemicals, of herbicide actives, was over 50 different active ingredients. So this just gives you some idea into the complexity of this issue when you talk about the behaviour of each of these chemicals in a range of different cropping soils, in a range of different environments, where there's different rainfall, different temperatures. You can start to appreciate that trying to determine how they all behave is a very difficult scenario to address. You can also see the different applications to the different crops here. So some you can see here, for example, atrazine is very specific to canola, triazine tolerant canolas, and you can see the higher rate of atrazine there. You can also see coming in here, this chemical here, pyroxasulfone, is a fairly new chemical under the trade name of Sakura, and in fact those applications only came from the final 3 years. So the other thing that we have to take into account is, over the coming decades, is the changing spectrum of herbicide use and how that might then impact on soil functions as well. Some other important chemicals here are the sulfonylureas, trisulfuron and metsulfuron methyl, the phenoxyacetic acids, or the synthetic auxins, MCPA and 2,4-D, and diflufenicin I'll also point out here as well, because it will crop up down the track. So I just wanted to talk a little bit about the next step in defining the exposure of herbicides in soil, and that's, once herbicides are applied, they generally all start to undergo breakdown. So we've moved away from the chemicals like DDT that will persist in soil for 10, 20 years. So generally speaking, most of the chemicals, of the herbicides in use today, will have a half-life, that is, half of the chemical applied will break down in less than 180 days. That's generally speaking, and most of them, I guess, probably would have a half-life of less than 60 days. So when we're talking about half-life over time, if we apply a chemical in January, we might see half it disappear by the start of February. So the half-life here would be 1 month, and so on. It will lose half of the remaining concentration over time until the next time that it might be applied in January, we might have none left in the soil. So that's one chemical. However, a different chemical might have a different behaviour in soil. It might have a slower half-life, that is, it's more persistent, and many of the pre-emergent herbicides that we use for residual weed control are designed to last longer in soil so that they control weeds for a longer period of time. However, what you can see here is we've got a bit of a wavy line. So not all herbicides, this half-life is just a generalisation of what actually occurs in soil. So how a chemical breakdowns depends a lot, in particular, on the temperature. So the warmer the temperatures, the faster it will break down. And also the microbial activity in soil, which is often driven by such things as water availability. So for example, if we have a rain event here, we can see that the concentrations decreasing a lot quicker. So yes, in particular, rainfall will drive herbicide breakdown and dissipation movements through the soil a lot faster than under dry conditions. Similarly, soils with higher organic matter generally have higher microbial activity, and herbicides will therefore generally break down a lot faster. One thing I just wanted to point out was, what if we have a chemical like glyphosate that we don't just apply once per year? Then it comes down to the frequency of application. So here you can see we're getting nice quick breakdown. However, we haven't quite degraded all of it by the time we have another application. Here we might have wet conditions at the start, but we might have a drying out throughout the season and have a really dry season. And you can see it's not breaking down as fast. And we might have another spray towards the end of October, for example, if we're spraying out the crop that didn't grow well because it had dry conditions, or if we have a Roundup Ready canola, for example, and then this could be the breakdown there. And then if we've harvested and we now have a fallow and we want to control weeds in the fallow, if we apply again. So what you can see here is that we're not actually giving it time to fully break down, and in fact we're getting this slow accumulation. So this is due to the frequency of application combined with the factors that determine dissipation and persistence. So I talked about glyphosate in that example, and glyphosate will crop especially used chemical in the media and the wider public. So what we also need to consider is that as chemicals break down, they will break down into smaller parts of the original molecule, sometimes called metabolites or breakdown products. And glyphosate is one example where the breakdown product AMPA, although not as toxic as the original parent molecule, does actually exhibit some phytotoxic attributes. So it does cause some impact on plant health, though definitely not to the extent of glyphosate. And what you can see here is a picture from a US soybean field, and it's a transgenic soybean, so it's Roundup Ready, which means it can resist the toxicity of glyphosate. However, soybeans can also break down glyphosate into this compound here, and this yellowing, what they call yellow flash, is actually due to the slight phytotoxicity of this breakdown product. So this breakdown pathway is also the main pathway by which glyphosate breaks down in soil. So soil microorganisms favour this pathway. So they'll break down glyphosate into AMPA, and then eventually they'll start breaking this in half, although this process for AMPA to break down is a lot slower, and you'll end up with just some phosphate and some ammonium, which will get converted to nitrate and then either be taken up and used by the microbes or plants as well, and the carbon will be given off as carbon dioxide. So we conducted a survey of 40 paddocks last cropping season. We took samples at the start of the cropping season, so in April, May, before any of the pre-emergent herbicides for that season had been put down. because we were just interested in getting a feel for which herbicides were hanging around or accumulating, potentially accumulating and being there at the start of the season before that season's application actually got put down. And what we found was that out of the 40 paddocks we surveyed, we actually detected the breakdown product of glyphosate, AMPA, in every topsoil sample that we took. This graph here says nothing about the concentration at which we found them in. It's purely just if we could detect them or not, which just gives you a snapshot of what we found. We also found glyphosate in most topsoils, so over 75%. And surprisingly, I guess, we also found a lot of trifluralin as well. What was also interesting is that these three chemicals generally considered to stay where they're put. Basically, they all bind very strongly to soil. The glyphosate and AMPA bind to soil minerals through this phosphate group. So they behave very similarly to phosphorus fertilizer. They bind quite strongly. Trifluralin, on the other hand, binds very strongly to organic matter. And generally speaking, where they are applied, they usually are retained, so in the topsoil. However, we did find them at the 10 to 30 centimetre depth, which suggests some kind of movement, whether it's through soil colloids, so very fine particles pulling the compounds down to the lower profile. In some cases, it could be some tillage or other disturbance, and in some cases, even in lighter soils with lower organic matter, the dissolved portion of the chemical may actually be moving down as well. We also detected quite a bit of diflufenicin, which also has a fairly long half-life. And although we detected 2,4-D, the levels at which we detected the 2,4-D were very low. So 2,4-D and MCPA, although, as you saw in the slide previously, although they're applied a lot in these cropping systems, their half-life is quite short, so they degrade quite rapidly. You'll also see that we detected more chemicals, generally speaking, in Western Australian systems, so lighter soil types, very dry summers. So when it's hot, it's dry, so that there's not much microbial activity. And when it's wet, it's cool, so again, there's not much microbial activity to push breakdown of chemicals. So just focusing in a little bit more on glyphosate and AMPA residues that we detected. So now we can move and talk about how much was actually there at the start of the cropping season. So this just gives an average representation of the glyphosate, which is the brown cylinders here, and the AMPA, the yellow cylinders, in the three different states where we measured. So there are about 10 to 15 paddocks from each state that we looked at. A full tank here represents a full tank of label rate application. So if there's a full tank down here, that means that there was effectively a full tank of glyphosate remaining in soil. So for example, in New South Wales, on average, the previous application of glyphosate had broken down, so there was only really less than a quarter of a tank of glyphosate remaining in soil. However, a full tank of glyphosate will break down to a full tank of AMPA, and then so on until it's completely mineralized. However, what we did see was that on average there is some accumulation of this breakdown product AMPA. And generally speaking, as we move to the drier climates, drier summers, and lighter soil types, we saw greater accumulation of glyphosate and AMPA. So that's on average. What about the paddocks with the highest amount? So in some paddocks in New South Wales, in one paddock in New South Wales, we found that half a tank of glyphosate had been remaining from the previous application. But what you can see here in South Australia and Western Australia on some paddocks, we actually found that there was more than one full tank of glyphosate in the soil, which means that there's carryover from at least the previous 2 applications of glyphosate, and even more so of the AMPA. So again, this is just talking about the exposure, not the toxicity, not the inherent toxicity of these chemicals. So the risk that they're posing to the soil functions and the crop production depends on both factors. So we really need to talk about the toxicity as well. Before I do that, here's just another quick snapshot of the other chemicals we found. And this is just one paddock in Western Australia, which had the highest total chemical load. And what we found in this paddock was that we're actually getting accumulation from one season to the next of trifluralin. So a full tank of trifluralin, which is usually only applied as a pre-emergent 1 year prior to the start of the 2015 season, remaining in soil, a full tank of diflufenicin, a fair bit of diuron, and then a few trace amounts of some other chemicals as well. So there's a real combination of residues there. And I guess what we don't know is how these chemicals might act in concert. So each of these has a different mode of action, so they'll all be impacting on different pathway in the plant. That's if they are indeed bioavailable. But what we don't know is whether together that will have an impact on the crop growth. All right, so that covers off a bit on what we now know about exposure in the soil, and we're going to follow up that survey with an even bigger survey in the coming cropping season through the National Paddock Survey, which is looking at 250 paddocks all around Australia. And this survey is looking at 2 zones within each of the paddocks, a high yielding zone and a low yielding zone. So hopefully what we will be able to do from this survey is look at any relationships between herbicide residue concentration in soil and crop performance, and put that together with many other factors that are being measured to try and work out what percentage of yield decline is being caused, if any, by herbicide residues. But moving now on to the toxicity side of things, I thought I'd start with earthworms because this is one of the toxicity measures that is most commonly measured when a herbicide undergoes registration in Australia and all around the world, in fact. So before they're registered, they have to undergo a few different toxicity tests, mammalian, fish, crustaceans, algae. But in terms of soil, really the only measures that are undertaken are earthworm toxicity, and they may also look at carbon and nitrogen cycling. However, it's quite difficult to get our hands on this data. We're still working with the APVMA, who register herbicides in Australia, to try and get more of this data that was presented at registration, which should be publicly available. In the meantime, this is some of the acute toxicity levels towards earthworms, and this here is a reproductive toxicity, and this is the no observable effect concentration. So, for example, glyphosate at 29 milligrams per kilogram has no observable effect on earthworm reproductive toxicity. Now, to put this in context, you can do a quick conversion from kilograms of herbicide applied to milligrams per kilograms of soil by multiplying by 3/2. So, for example, if I put 1 kilogram per hectare of glyphosate on, I'd expect in the top 5 centimetres of soil to have 1.5 milligrams per kilogram. So in terms of our residue survey, we're not yet getting close to this. The highest, I think, detected levels we reached were about 2 milligrams per kilogram. And if another application of glyphosate were to go on top of that residue that was already in soil, you might reach immediately after application 4 milligrams per kilogram or 5 milligrams per kilogram. And that's only in the top 5 centimetres of soil. So generally speaking, the levels of herbicide that you would need to get to cause toxicity to earthworms is really quite high. And really none of the herbicides that we've measured reach anywhere near these levels. And I guess that fits with a lot of farmer anecdotal evidence that I heard when I was out sampling, in that since they've switched to minimum tillage, although they've increased in the use of herbicides, they've actually started to see earthworms come back into their systems, which weren't there prior to prior to switching to minimum tillage. So we must keep in mind that other farming practices, for example tillage, although it's not chemical, it's a mechanical disturbance, can actually have as large or even larger impacts on the soil biology and function as other inputs into the soil. So that just gives some context on earthworm toxicity. On now, I just wanted to focus a bit more, follow on with the glyphosate, because glyphosate's a very well-studied chemical, which gives us enough data to look at its impacts on soil function, on soil microbial function. And we recently published a paper where we collated as much information as we could find about the impacts of glyphosate on soil microbial biomass and activity, measured as soil respiration. And we tried to put all these different studies together and statistically analyse them using this meta-analysis techniques to tease out overall at label rates used in agricultural systems, are we having a negative impact on microbial activity and microbial biomass? And what we found is that label rates are usually equivalent, as I said before, to about perhaps 2 milligrams per kilogram. We set a fairly high threshold here of 10 milligrams per kilogram as our label rate. And what we found was that there was no significant increase or decrease of microbial activity on microbial biomass. So in this graph, data points above 1 mean that when glyphosate was applied, there was a positive effect on microbial activity or microbial biomass, and if they're below 1, there was a negative impact on biomass or activity. So overall, on average, there was a slight decrease in microbial activity, but the variation between studies was so great that it was not really significant, and in fact often the decrease was observed in the first 7 weeks, and after that we would see an increase in microbial activity. So many disturbances will exhibit that kind of behaviour, soil disturbances. So any soil input of chemical or a mechanical disturbance, you'll often see a slight dip and then an increase as the microbial community rearranges itself. Interestingly, at very high rates, which you would not find in agricultural systems, but you might find after a spill, we actually saw statistically significant increases in microbial activity and microbial biomass. And it's well known that many species of organisms in the soil, microbial species, can break down glyphosate, and in doing phosphorus in the glyphosate molecule as a food source. So at levels this high, the inputs of phosphorus and nitrogen from glyphosate actually represent a kind of nutrient source, but definitely at this level it's so low that it's not going to make too much difference. There's also been some other reviews done looking at the impacts of glyphosate on plant mineral nutrition, rhizosphere, microbiota, and the interaction of glyphosate with plant disease. So a few studies here and there suggest that there is an interaction of glyphosate application with increases in plant disease, but this review, which I would recommend if you can get your hands on, is quite a comprehensive review, suggests that most of the available data supports the view that neither the glyphosate-resistant transgenes nor the glyphosate itself increases crop disease. However, there may be some effects of glyphosate on mineral nutrition or disease under very particular but uncommon conditions. So in that case, it's not widespread, but under some scenarios it could be a site-specific impacts. All right, I just wanted to have another quick look at the other herbicides that we focused on in our study, and the other commonly used toxicity threshold is looking at the nitrogen cycle. So specific organisms in the soil mediate or facilitate this cycle here. So nitrogen fixation from the atmosphere and in the soil. We also have fertilizer and manure inputs, for example urea, and that organic N is then converted through to ammonium by microorganisms, and then a specific subset of microbes actually converts the ammonium through to nitrate in the process of nitrification, and then denitrification causes the loss of nitrogen from the system in gas. So we can apply herbicides in laboratory conditions and monitor these levels and work out if any of these pathways are being impacted by herbicides. This is just some example data that we have from the lab that we're now putting together to rank the herbicides and on their inherent potential to affect the nitrogen cycle. In this case, this process here, nitrification, which we measure by measuring the nitrate concentration over a 28-day period. So under control conditions, we get a conversion to nitrate, and for example, in this soil we had just over 185 milligrams per kilogram of nitrate produced. Glyphosate at 1-time label rate and a 5 times label rate had no significant impact on that process. However, diflufenicin, another of the commonly detected residues, did actually inhibit that process. And this here is our positive control. It's a fungicide, which we use because we presume that being a fungicide, it will have impacts on the microbial community. However, what you can see is that the loss of function is probably less than 10%, so less than 10% inhibition of that function. And for registration of these chemicals in Australia, I think the threshold is generally about 20 or 25% loss in function. So although there may be some impacts at these levels, at a 1 times label and a 5 times label application rate, it doesn't cross the threshold of 25% inhibition, and registration is therefore allowed. So I guess the question there is what in terms of crop production is actually significant, and if these organisms that carry out this function are just an indicator species for total soil processes, what thresholds are we willing to accept? And what we're trying to do, I guess, is provide a ranking of the different chemicals and show these actual thresholds so that then a farmer can make their own decisions. Now, I just wanted to talk, aside from the impacts on soil function and soil organisms, herbicides being herbicides do have direct impacts on the plant itself, and that's what they're designed to do. And although many crop species have a higher threshold than the weeds, it's actually the dose that determines the toxicity. Glyphosate is generally accepted as being inactivated when it reaches the soil through that binding mechanism I mentioned earlier, where the phosphate group binds to soil minerals, and it's no longer available for plant uptake. So it's considered as a contact herbicide when it hits the plant leaves. But what we wanted to do was follow up on some research that has been conducted in Europe, and there was also some early research done in the 90s in Australia that showed that under some conditions, glyphosate residues in soil can actually be remobilized and therefore be taken up by crop plants, weeds as well, but also crop plants, and cause some plant-back damage. So we chose a soil from Western Australia. It's a light sandy soil that has a low phosphorus buffering capacity, and the phosphorus buffering capacity is what regulates glyphosate bioavailability in soil. And we did a small bioassay where we grew the plants for 6 weeks and measured biomass. So the glyphosate was applied 1 month prior to sowing the seeds, and it was aged in the glasshouse under relatively moist conditions, so there will have been some binding and some breakdown of the glyphosate during that 1 month. These are the glyphosate application rates, and you can see without any added phosphorus, the threshold at which we started to see 10% biomass loss, we determined or estimated through modelling this decrease here. You can definitely see an impact here. But our model suggested 12.5 kilos a hectare was needed to be applied to soil to have any impact. And so label rates being 1, maybe you'd get up to 2 kilograms per hectare and not going to have any impact. And in actual fact, there's this effect where you do see slight increases in plant growth at low rates, and there's a number of scientific papers on that as well. However, what we were more interested in is what happens if you apply phosphorus fertilizer. So phosphorus fertilizer can compete for the binding sites of the glyphosate molecule and make the glyphosate become more bioavailable in soil. And the same conditions, and what we found here was that the threshold was now brought back down to about 3.5 kilos per hectare of glyphosate applied 1 month before sowing. So based on what we found in the soil survey of 2 milligrams per kilogram kind of was the maximum level detected. If you were to apply another kilogram per hectare application up front at sowing, which some growers do just prior to sowing, you might be pushing up towards this threshold here. And I guess these plants were grown under well-watered, non-stressful conditions. So I guess where we want to look at next is what if there are other stresses? Will this sublethal impact of low levels of glyphosate contribute to increasing the stress of the plant and therefore its ability to fight back against those stresses? So that's where we're at the moment, and stay tuned, there'll be more work done there. In terms of the other residues that we found and their direct impacts on plant health, what we struggled to find is toxicity thresholds for soil herbicide residues and their direct impacts on crop plants. The only thing we could find of interest was to do with trifluralin and their suggestions that there's can be damage to oats at 0.1 milligrams per kilogram, and we found 4 sites exceeding those topsoil concentrations prior to sowing and prior to pre-emergent herbicide application, which means that growers may have then gone in with a trifluralin application for that year. So that may be pushing up towards the thresholds there. And wheat, which is less sensitive than oats, we found a number of sites in Western Australia above this threshold as well. So again, we need to do some more work in the glasshouse to really define those thresholds. These values were taken from a paper from USA, but again, it will depend a lot on soil type and how strongly these residues are bound to the soil, so whether or not they're actually available for the plant to take up. So just to wrap up in terms of direct impacts on plants and plant back, based on talking to a number of farmers and agronomists, we think there's probably more concern about direct impacts on the crop from herbicide residues, so plant back impacts rather than impacts on soil function. And these are just some screenshots from Twitter that I got from a number of different agronomists and growers that just demonstrate, for example, here, this agronomist, Simon Mock, says soil type matters with lentils, and then I think his client probably replies here, are they ours? We didn't accidentally poison them again, did we? And he replies, it's probably a combination of herbicide damage, dry frosty start, loose and flea, and late rolling. Even us agronomists can get it wrong. So what I wanted to highlight here is that it's very difficult to attribute any yield decline or plant back damage or poor emergence directly to herbicide damage. There's no quick test that you can do like a nutrient leaf test that can tell you that it's a particular herbicide that's done some damage. And from soil type to soil type, those thresholds are going to vary, so we really need to start teasing that out. And here again, sulfonylurea herbicides are something of real concern to a lot of growers. I guess sulfonylurea herbicides, IMI herbicides, which are both group Bs, and also Lontrel or clopyralid, which is a synthetic auxin, are the 3 big herbicide classes that that growers are worried about in terms of plant back damage. And in fact, one agronomist from Vic Department of Primary Industries, or DEGITA, suggested that in the Mallee and Wimmera last season, the pulse crops that were growing down there, he thought that 20 to 30% of paddocks were actually suffering from plant back damage from clopyralid because of the dry season that had before and the slow breakdown of that chemical, of which herbicides, legumes are very sensitive to. So in conclusion, the work that we've done so far, we found that glyphosate, AMPA, trifluralin and diflufenicin were frequently detected in cropping paddocks, and there's some evidence for accumulation from season to season. And that could be a combination of the frequency of application and also the inherent persistence. So the frequency of application for glyphosate and AMPA, but probably the persistence more so, the inherent persistence of trifluralin and diflufenicin, and especially dry seasons, which means that their breakdown is a lot slower. So that's not to say that in every season there will be carryover. If we have a good wet season, a lot of rainfall, then usually these chemicals will not carry over. So then it comes down to how frequently they're being applied. And in general, we think that there's a low risk to soil biological processes when using label application rates and given sufficient time for these chemicals to break down. And that's based on a number of literature studies that we've done, and we've published that in an Advances in Agronomy manuscript. Probably of more concern to growers is this issue of direct impacts on following rotation crops and herbicide carryover. So we need further research to define critical thresholds to avoid those potential negative impacts on crop production. But for the time being, we really need growers to keep accurate spray records. So if there is damage, we can trace that back. We think that rotating herbicide classes, so not consistently reapplying, for example, a sulfonyl urea, which will result in accumulation and therefore increase the potential for damage. So rotating herbicide chemical classes, and stay tuned, because we are trying to develop a number of tools over the next couple of years. First and foremost, we want to try and develop a handheld app for farmers so that they can put in their application records and their weather conditions and some rudimentary soil parameters, for example pH and texture and organic matter, and really give them clearly defined breakdown timelines so that they can start to get a better understanding of how the herbicides they're using will break down from season to season and paddock to paddock. Because at the moment the half-lives or the breakdown parameters that are given to farmers are very broad and very general, so it's often just one number. For example, glyphosate has a half-life of 30 days, or don't plant a legume after metsulfuron application for 180 days. But clearly there's a huge range in potential persistence over the different soil types and climates. So we want to really come up with better tools for farmers to better understand that. We also want to come up with rapid tests that farmers can use to diagnose herbicide residues in the field. These are similar to pregnancy test kits, and they can be conducted in 30 minutes, and they're a lot cheaper than laboratory analyses, which can range anywhere between $100 and $500 per sample, whereas these will probably retail around $20 per sample. And like I said, you can do it in the field, scoop of soil, shake it up, put a drop of the extract on these, and if the herbicide is present, for example here 1,000 milligrams per kilogram, the test line disappears. If the herbicide's not present, then test line will appear, and the threshold here is set to about 50 ppb. So above that level, sorry, this is for atrazine, then you'll get this line disappearing. So a farmer will know, yes, there are atrazine residues that may potentially damage my crop. So that they will know that within a matter of 30 minutes. So we want to try and develop more of these kind of kits. And just finally, as I said before, not knowing if a yield decline or a poor germination is due to a herbicide or some other reason, I think is really something that plays on farmers' minds. You know, was it because of that herbicide? And I'll never know. Or was it because of frost? Or was it because of manganese toxicity? Or was it because of deficiency? So we want to try and develop leaf tissue testing tools that can give us a definitive answer as to whether a crop is being damaged by a particular herbicide. And we know the herbicide, the biochemical pathways in the plant that each herbicide impacts on. For example, glyphosate acts on this shikimic acid pathway, and it blocks it here. So normally you'll have similar levels of both these metabolites in plant tissue, but if you block that pathway, which shikimic acid is converted to this amino acid, then you'll have a build-up in shikimic acid because it can't get through that enzyme to make the phenylalanine. So we'll see this signature damage, and we want to come up with tests, leaf tissue tests, for all the different classes of herbicides so that we can then tell farmers yes or no that damage was indeed caused by that herbicide. So I'll wrap it up there, but happy to take any questions, and I'd just like to acknowledge GRDC and all the farmers and farmer groups that we've worked with, the soils unit at Wollongbar DPI for all their support, and the project team members in University of WA, Southern Cross University, University of Adelaide, University of Sydney, Vic, Degida, DAFWA and QDAF. So thank you very much.
Thank you, Mick. That was chock full of information. Getting on to the question, we have a question from Adrian Hayes. Were the samples taken from paddocks used in irrigated cropping?
No, actually all those samples were taken from dry land crops. So we would expect that irrigated crops, the herbicide breakdown would be a lot faster due to the moist conditions. But no, all those samples were taken from non-irrigated crops. Yeah.
Good question. Luke says he's got a question.
Yeah, thanks, Mick. Look, it's just a simple one. When you came to your soil pregnancy test, when you said the dollars per sample you're hoping for, the sound cut out just as you said the dollars. Can you tell us what that estimate was?
Yeah, we think probably around $20 per test. So that might actually include a couple of test kits as well. So per sample it may even be cheaper than that, but it's difficult to know. So we're working with a commercial partner there to develop those tests. So I guess at this stage we're road testing them really in this project, and hopefully they'll be available soon, because I know a lot of agronomists that I talked to were very interested, particularly if they could get a test for the IMI herbicides, which are being used more frequently now with Clearfield technology, so crops that are tolerant to the IMI herbicides. And the IMIs have a longer half-life, so they persist a lot longer than for example, glyphosate or even atrazine for that matter.
Okay, thank you. There's a few more questions that have popped up here. One from Jeff Casburn, and he says the herbicide buildup is surprising. How much of an issue could it become in crop-only paddocks? Yeah,
so I think that was probably partly reflected in the higher levels that we detected in Western Australia as well, because most of those were continuously cropped paddocks. So I think really we're probably looking at more of an issue in those kind of Mediterranean climates. So the dry summers, I think, are probably a real issue, because I guess in New South Wales and Queensland, having wet summers, when it's warm and when it's wet, that really facilitates the breakdown of those residues. So I think the more Mediterranean climates and the soil types that are a lot lighter and lower in organic matter are where the issues of accumulation or residue buildup were seen. But as I said, it's going to change from year to year, and it's not to say that they will always accumulate. And in fact, for example, if you then switched to a pasture and really backed off on the herbicides, then those residues over the next couple of years would degrade, and especially what we see in our laboratory testing of soil functions is that the microbial functions bounce back very quickly as the herbicides degrade. So often even if we do see an impact of the herbicide application, it will only be short-term impact.
Another question here from Harry Kibler, who said that they apply molasses after applying glyphosate to increase microbial activity, and they grow native perennials. Would you care to comment on that, Mick?
Yeah, well, that's really interesting. I haven't actually had any experience with that, but yeah, I would guess that by putting molasses on it would stimulate that microbial activity, and in particular, we do know that there's been some work done that shows that when the carbon to nitrogen ratio or the carbon to phosphorus ratio is higher, then organisms will actively try and break down the glyphosate to get at the N and the P. So it seems to break down faster. So I'm just guessing here. I mean, I haven't done any research on this area, but I would imagine that it would actually break down a bit faster.
Steve Kimber has raised his hand.
I just wanted to make more of a comment than a question. And when Mick showed the field survey data, it's important to note that that's total residues, not necessarily bioavailable residues. And I suspect that where there are very high residues, it's because probably the residues are not really bioavailable. But in the lab, we use a very aggressive extraction technique. It's important to note that they, as shown in the glyphosate pot trial, they have the potential to become bioavailable, as in glyphosate when you add phosphorus to the soil.
Okay.
Yeah, thanks, Steve. Yeah, that's spot on.
Thank you, Steve, for that comment. I'd just like to take this opportunity to thank Mick again for presenting not once but twice for the SNOC webinar series, and I hope that everyone got a lot out of it. There's a lot of information in there, and we look forward to continuing to follow the project